Hey everyone, and welcome back to another explainer. We walk past plants literally every single day. Maybe we occasionally stop to admire a nice flower or grab a piece of fruit. But hidden right beneath our noses, well, hidden at a microscopic level, is this incredibly high-stakes world of survival, chemical warfare, and frankly, some absolutely mind-blowing engineering. So today, we're going to decode the fascinating and sometimes ruthless mechanics of sexual reproduction in flowering plants.
Okay, let's dive into this. To track this biological hero's journey, we're going to follow a chronological four-step roadmap. First, we'll look at preparing the gametes. Second, the art of pollination. Third, the double fertilization climax. And finally, seeds, fruits, and beyond.
But before we get into the nuts and bolts, or the pollen and pistils, we have to acknowledge the ultimate biological problem here. Think about it. If you're an animal, you can walk, fly, or swim to go find a partner. But how exactly do you find a mate when you are permanently rooted to the ground? This extreme immobility is a massive hurdle, and it requires some ingenious evolutionary solutions to ensure a species doesn't just survive, but actively thrives.
Which brings us to step 1, preparing the gametes, or as I like to call it, building the biological fortresses.
Starting over on the male side of things, we have the mature anther, and it basically acts as a multi-layered, heavily guarded fortress. Beneath those outer protective layers, like the epidermis, lies a highly specialized layer called the tapetum. Now think of the tapetum as a dedicated, high-energy kitchen. Its sole purpose in life is to provide dense nourishment and these really unique compounds, like sporopollenin, to the developing pollen grains that are growing safely tucked inside the microsporangia.
Deep inside that protected zone, an incredible transformation kicks off called microsporogenesis. Basically, a single sporogenous cell steps up to the plate to become a microspore mother cell. Through the process of meiosis, this one mother cell actively forges 4 distinct microspores. They're initially clustered together in what's called a tetrad, but once they mature and separate, boom! You have your adventurous, tough-as-nails pollen grains, totally prepped and ready to brave the outside world.
Meanwhile, the female structure is busy preparing an even more secure fortress. What's really interesting here is the design of the typical anatropous ovule. It's completely inverted. Massive protective layers called integuments wrap tightly around the precious embryo sac inside. They leave just one tiny strategic little doorway open at the bottom, which is called the micropyle. And this inverted design, absolutely not an accident. It protects the vital contents while creating a highly controlled single access point.
Now, deep within this ovule, megasporogenesis is taking place, and honestly, it is a brutal game of survival of the fittest. A megaspore mother cell divides through meiosis to create 4 megaspores. But in a totally ruthless move to ensure maximum resources are kept for the winter, 3 of them just degenerate. They die off. Only a single, dominant, functional megaspore survives to actually carry on the legacy.
And that sole surviving megaspore, it goes into overdrive, undergoing 3 rapid rounds of free nuclear division. This results in one of the most famous trivia facts in all of botany. The classic polygonum-type embryo sac reaches a 7-celled, 8-nucleated stage. It organizes itself just perfectly. You've got 3 antipodal cells at the top for nutrition, a massive central cell with 2 polar nuclei, and down at the bottom, guarding that micropyle door we talked about, is the vital egg apparatus, complete with 2 little helper cells called synergids. Moving right along to Chapter 2, the art of pollination. The gametes are ready and waiting, so now it's time to find a match.
When it comes to matchmaking, plants actually have a few options. They can keep it super close to home through autogamy, which is just self-pollination within the exact same flower. Then there's geitonogamy, which is kind of a weird middle ground. This is where pollen moves to a different flower, but it's still on the exact same plant. So genetically, it's self-pollinating, but ecologically, it requires a courier to move it, making it cross-pollination in practice. But the absolute gold standard for genetic diversity, that's xenogamy. True cross-pollination between entirely different plants.
So to achieve that true cross-pollination, plants enlist an absolute army of careers. They use abiotic or non-living factors like the wind, literally producing millions of lightweight, non-sticky pollen grains just hoping the breeze catches them. Or occasionally, they'll use water. But the real magic happens with biotic careers. Plants actively manipulate the animal kingdom. They enlist birds through a process called ornithophily, or insects via entomophily. And they do this by straight-up bribing them with sweet nectar rewards, edible pollen, and irresistible fragrances just to get them to do their bidding.
And this brilliantly illustrates the sheer lengths plants will go to Just look at these mechanical strategies. For wind pollination, the Arctica plant uses a literal gunpowder mechanism to violently explode its pollen out into the breeze. For insect manipulation, the salvia flower uses a lever mechanism. An insect lands to get some nectar, accidentally trips a lever, and smack! It gets whacked on the back with a perfectly placed dose of pollen. Or consider the ficus plant. It uses a literal trapdoor to lock specific wasps inside its fig until the pollination job is done. Nature is wild.
But here's the catch. If you're going through all this trouble to cross-pollinate, you definitely don't want to accidentally self-pollinate. So plants have built-in fail-safes. Some are invisible genetic safeguards, like self-sterility. If the S1 or S4 genes in the pollen match the stigma it lands on, it is instantly rejected, no growth allowed. Others use physical blockades, which is known as herkogamy. The calotropis plant, for instance, packages its pollen into these heavy little bags called pollinia that literally burst outward. This makes it physically impossible for the plant to pollinate itself without a very strong insect coming along to lift those bags away.
All right, Chapter 3, the double fertilization climax. The pollen has officially landed, setting the stage for a mind-bending botanical event that is entirely unique to flowering plants.
Because you see, just landing on the stigma isn't an automatic win. It's really just the start of a strict, incredibly rigorous chemical interview called pollen-pistil interaction. The female plant actively vets the pollen. If it passes the test, the pollen grain sprouts a tube that literally has to digest its way down through the solid tissue of the style. It's navigating a microscopic obstacle course, guided entirely by a chemical gradient of calcium, boron, and inositol that basically points a neon sign saying, ovary this way.
Now, this hollow, digested tube is carrying highly precious cargo, 2 male gametes. It snakes its way all the way down, finally reaching that tiny little doorway we saw earlier, the micropyle. Right inside that doorway, those helper cells, the synergids, have special finger-like projections called the filiform apparatus. Think of them like tiny botanical air traffic controllers, safely guiding the pollen tube right into the embryo sac so it can release those 2 male gametes.
And then we get the grand finale, double fertilization. It's a biological magic trick, guys. The first male gamete fuses with the egg cell, creating the zygote, which becomes the baby plant. But what about that second male gamete? Well, it fuses with the 2 polar nuclei sitting in the center to create a triploid primary endosperm nucleus. So in one fell swoop, the first sperm creates the baby, and the second sperm creates a massive, nutrient-dense packed lunch called the endosperm to feed it.
And that leads us to our final chapter, seeds, fruits, and beyond. The flower's main job is done, so now we transition to the legacy it leaves behind.
So about that packed lunch we just mentioned, the endosperm, plants actually handle this food stash differently. During embryo development, some plants, like most monocots and castor plants, are pretty conservative. They retain a large portion of that endosperm inside the mature seed for later. We call these endospermic seeds. Others, like most dicots, are totally gluttonous during development. The growing embryo completely consumes the endosperm right away and transfers all that stored food into its own fleshy seed leaves, the cotyledons. These become non-endospermic seeds.
Ultimately, the entire protective structure completely transforms. That protective ovule hardens into a tough seed coat safeguarding the embryo inside. Meanwhile, the surrounding ovary wall swells up and ripens into a fruit. But sometimes plants like to hijack their own system. A true fruit, like a mango, develops purely from the ovary. A false fruit, however, like an apple, actually recruits other parts of the flower, like the thalamus at the base, swelling it up to become the fleshy, edible part that we eventually eat.
Now, we've seen the absolutely incredible lengths plants go to for sexual reproduction. I mean, the meiosis, the complex lever mechanisms for pollination, the wild magic trick of double fertilization. But I want to leave you with a really fascinating biological loophole. What if a plant, after evolving all of these complex systems over millions of years, just figured out a way to fake it? Well, a phenomenon called apomixes exists in plants like certain grasses, where they completely bypass fertilization altogether. They literally mimic sexual reproduction to produce a viable seed that is a perfect 100% genetic clone of the mother plant. No pollen, no partner, no problem. It really begs the question, in the relentless chess match of evolution, is the ultimate winning move sometimes just flipping the board altogether? Think about that next time you see a field of grass. Thanks for joining me, and I'll catch you in the next explainer.